In plant cells, chloroplasts carry out photosynthesis. These large, dedicated organelles contain a variety of membrane components that convert the energy in light into the energy carriers NADPH and ATP, which in turn fuel the production of sugars and other molecules required by the cell. Chloroplasts have three distinct membrane systems: a two-membrane envelope akin to that surrounding mitochondria, and the internal thylakoid membrane system. Within the thylakoid membranes, large antennae consisting of hundreds of light-absorbing chlorophyll molecules capture light energy. When a chlorophyll molecule absorbs light, the energy bumps from one chlorophyll molecule to another until it passes to a special pair of chlorophyll molecules in the reaction center of photosystem II. In the reaction center, the energy causes an electron in chlorophyll to jump to a higher energy level. This jump initiates a long series of electron transfers. First, a neighboring molecule accepts the high-energy electron. In the meantime, another neighboring molecule donates a low-energy electron to the deficient chlorophyll molecule. In turn, this donor molecule receives a low-energy electron from water. After this series of transfers occurs 4 times, 2 water molecules are split into 1 molecule of oxygen gas and 4 protons. The photosystem shares the thylakoid membranes with an electron transport chain. When light bumps an electron out of the photosystem, the electron is removed by a small diffusible carrier molecule. As shown before, water replenishes lost electrons. The diffusible carrier molecule brings the electrons to the cytochrome B6F complex, which uses part of the electron's energy to pump protons across the membrane. From there, the electrons travel to photosystem 1. Just like photosystem 2, photosystem 1 absorbs light through its own antenna system and kicks electrons to an even higher energy level. After 2 such high-energy electrons have been produced and delivered to ferredoxin NADP reductase, they drive the reduction of NADP to NADPH. To liberate a molecule of oxygen from 2 molecules of water, the cycle must occur twice. To make the system work, each member of the electron transport chain has to be finely tuned to have an appropriate tendency to receive or donate electrons, measured as a redox potential. When photosystem 2 is excited by light, it has a high tendency to donate electrons. The next component is more likely to receive electrons. The loss of an electron from photosystem 2 now makes it an excellent electron acceptor, receiving electrons from water. The next series of carriers in the chain make better and better acceptors, drawing the electron through the chain. The released energy is used to generate a proton gradient that fuels ATP production. To produce another high-energy electron, photosystem 1 must also absorb a photon of light. This second energized electron has an even higher energy level than the first and can pass to ferredoxin NADP reductase. 2 such electrons will produce a molecule of NADPH. For every 2 water molecules split by photosystem 2, 4 electrons are donated to produce NADPH.